the light-independent reactions. These reactions are happening continuously inside the cell, and they don't require any light. Independent reactions. Previously, they were called the dark reactions, and some of the older folks might still call them the dark reactions, but they changed it because the implication means that it only happens in the dark. And that's not true. It happens all the time because it's just biochemical stuff going on. As long as the chemical precursors are present, these reactions continue. So they happen whether it's light or they don't care if it's light or dark because they don't need any light to function. Okay. So the starting point of this, of course, is our old friend carbon dioxide. So 3 carbon dioxides. Now here's the interesting thing. They kind of go one at a time through the cycle. So as we do this, we have to kind of think of it as the net result is 3 carbon dioxides will be cycled through to make this happen. Okay? But the problem with the cycle is it doesn't just do everything all at once. It goes 3-2. So one at a time. These guys go through the cycle. When they go through the cycle, they encounter a very important enzyme. This is the only enzyme that we are going to remember. I'll write him in green, and his name is Rubisco. Rubisco. He doesn't have an A's ending, so he's a little bit strange. So that's the enzyme that is going to be involved in this process, and it's quite an important enzyme. And we'll talk more about Rubisco later. Right now, we just want to see the molecular process. And because it's a cycle, this is a cycle, it's actually called the Calvin cycle, right? Maybe we'll put that up here. Calvin cycle. Wow. Calvin cycle. It's kind of like the Krebs cycle. Calvin was the guy that discovered it or identified it. And so it's a cycle. So there's another molecule that's coming in here. And along with the presence of Rubisco, it's going to combine with the carbons and it's going to produce what's called a short-lived intermediate molecule. What's important is that it has 6 carbons. So it's a short-lived intermediate. That's what it's called. The reason we don't worry about it is because it's short-lived. It forms and then instantly breaks apart to produce a new molecule. But it does have 6 carbons in it. So what we're going to be doing is we're going to be tracking the number of carbons, right? It's a 6 carbon. So 3 of those carbons came from carbon dioxide. Or actually, 1 of the carbons came from carbon dioxide because it's happening 1 at a time. And the other 5 came from the molecule on this side, which will be the end of the cycle. So we'll talk about that in a minute. This immediately breaks apart into 3-phosphoglycerate. And it makes 2 of them, right, for each time. So if we're going to keep track of numbers, I'm thinking we should just do 1 carbon at a time. Why don't we just erase this 3 because it's going to confuse us. Let's just put carbon dioxide here and not worry about the 3. You'll see why we need the 3 in a minute, but let's just do it this way. Okay, then this makes 2 molecules. It splits into 2. Oops, I got to get my pen back. Splits into 2 3-phosphoglycerate molecules. Phosphoglycerate. Phosphoglycerate is a 3-carbon molecule. Okay, and we can actually draw it. We'll actually draw phosphoglycerate underneath here. Well, actually, let's not worry about the drawings. Let's just keep track of carbon molecules rather than we know what a phosphoglycerate looks like because we've seen it before. Okay? Now then this is going to turn into another molecule. But at this point there has to be an addition. This is going to turn into 1,3-bisphosphoglycerate. You might notice a lot of these molecules are the same ones from the citric acid cycle, kind of in reverse. if you look at glycolysis and you go backwards, you remember bisphosphoglycerate from glycolysis, right? So you'll see that it's almost the same molecules, but everything's kind of going backwards because we're starting with CO2 and we're building glucose. So 1,3 bisphosphoglycerate. And it is also a 3-carbon molecule, so we'd have 2 of them that would be formed here. 3-carbon molecule. But in order for that to happen, you can see what's happened. We've added a phosphate. Okay? You might ask where the phosphate came in this first one. It came from the other part of the cycle. So you'll see when the cycle's done that the phosphate in the first guy here, in the 3-phosphoglycerate, he was already there because he was part of what enters into this from the other side. But now we're adding a phosphate, which means, guess what? We're going to have to take an ATP molecule. And in fact, because there are 2 of them, we're going to have to take 2 ATPs, and we're going to have to turn them into ADPs. And the phosphate, the inorganic phosphate that we would normally add here, is now attached to the molecule as a bisphosphate. It has in the 1 and 3 positions. Okay? Then this molecule is turned into, let's go, oops, black. I thought I went to black there. This molecule is then turned into, let's go here. That's glyceraldehyde 3-phosphate. Remember that guy? Glyceraldehyde 3-phosphate. I have to write this on 2 different lines. And in order for that to happen, there has to be, this is also a 3-carbon molecule. In order for that to happen, there has to be another sort of process. We have to sort of add some electrons to this because we're building, right? So we're going to be adding electrons to create new bonds to build bigger molecules. And guess who our friend is? NADPH, which was made by the light reactions, can come along here and it can give the electrons to this molecule and it can produce NADP plus and H plus, of course. So the electrons have been transferred to create new bonds. And actually there would be 3 of those happening too. It would take, no, 2, sorry, 2 of those, because there's two biphosphoglycerates, so we should put 2 here. 2, 2, and 2. It's a little sloppy, but 2 of them are going to be used up in this process. Then, here's what's interesting. The glyceraldehyde 3-phosphate can follow 2 different paths at this point. Okay, I'm going to have to move this down a bit. And this is why you have to go around the cycle 3 times to make this happen. So you get this bisphosphate, this bisphosphoglycerate building up. 1 carbon goes through, builds it up. Another carbon goes through, another carbon goes through. After 3 carbons have gone through, you end up with lots of this glyceraldehyde 3-phosphate. You end up with 6 of them. And out of the 6, 1 of them is going to basically go this way and is going to follow the reactions of glycolysis in reverse and be converted into glucose. Okay? So that's where the glucose comes from. Now this arrow, I want you to make a note here. I can't write it in on my picture. There's not enough room. But if you go back to glycolysis, glyceraldehyde 3-phosphate was the end product of the first half. So you can easily see how those reactions could all be reversed to produce glucose, right? So this arrow stands for all of the reactions that happen in the first half of glycolysis backwards. So there's a lot going on there, but we're not going to write all those in. So that's how the sugar gets made. Now that's 1, right? The rest of them, after 3 spins through, The rest of them go this way and produce ribulose biphosphate. Ribulose bisphosphate, not biphosphate. Ribulose bisphosphate. And of course, in order to do that, we've got to add some phosphate. And so we have to add ATP again. So we get our ATP coming into this process and adding its phosphate, phosphorylating this molecule so that it becomes ribulose. Maybe I should make that arrow go a little bit further, eh? Make it black, like that. I can slow a little bit, yeah. But I'm also watching the clock. Speed it up. All right, so here we have the ribulose bisphosphate, the ATP here. Now, we're going to put the numbers in at the end because we have to sort of bear in mind that this is a bit of a tricky process. Now, the ribulose bisphosphate, of course, is basically connects to this arrow, and that's the piece that, that's a 5-carbon sugar, and that's the piece that joins with the carbon dioxide again to make the cycle go through again. 5-carbon sugar. Glyceraldehyde 3-phosphate is obviously a 3-carbon sugar. Okay, so what happens here in terms of numbers? If one carbon dioxide goes through, you can see how it'll add to the 5-carbon ribulose bisphosphate and produce this 6-carbon short-lived intermediate, which breaks into 2 phosphoglycerates and produces 3-carbon molecules. And that turns into the bisphosphoglycerate, 2 of them. But in order for the process to continue, we need to have 6 glyceraldehyde molecules. We need to have 5 of them that will continue this way in the process, and 1 of them that continues along the path to glucose. So only 1 is going to get pulled out of here to turn into glucose, while the rest are recycled. So how do you get 5 if you only had 2? So what actually happens is there's sort of a buildup of bisphosphoglycerate that has to happen. So I'm going to go back now and I'm going to change some numbers. What really happens is the cycle goes through this 3 times. So there are 3 carbons. 3 carbons would produce, joining with 5 ribulose biphosphates, would produce 3 short-lived 6-carbon sugars, right? So 6-carbon intermediate. So there end up being 3 of those in total, but they'd be made 1 at a time in the cycle. That's the problem. And then that would mean they would break into 6. Total would be 6 here. So we can do the accounting, realizing that these 6 are not all made at once. They're made as a process of the cycle going through 3 times, which would also mean we would have 6 of these. 6. Okay? And now those 6, out of those 6 that would be built up as this happens, 1 of them will go to become glucose and the other 5 will go back to finish the cycle. So the cycle runs basically 3 times to get 1 glucose molecule made. That's kind of the way you should remember it. And it requires 3 glucose molecules. Now, actually, that's not even true. Let's think about the carbon numbers. 3 carbons would go in. Question? No, this would be a total of 6 here. Yeah, that's a good point. So let's add that as well. There'd be a total of 6 of those, 1 of which would go on to become glucose. But here's the problem. Here's the problem. 1 of them can't become glucose. It's only a 3-carbon sugar. So what has to happen is the cycle works in terms of 3 carbon dioxides at a time. So the cycle has to go around 3 times to get 6 glyceraldehyde 3-phosphates. Got it? Okay? The cycle has to go around 3 times to end up with this 6 right here on the glyceraldehyde 3-phosphate. Does that make sense? You guys having the same problem? Yeah, so if it goes around once, it only if it goes around once, you can trace the carbons, right? One carbon dioxide will make, with a 5 carbon sugar, will make a 6 carbon sugar. Then that'll be split into 2 3 carbon sugars and so on. But in fact, that's just keeping track of the carbons. What actually happens is, in order to get a 6 carbon glucose, if you put 3 carbon dioxides through, then the cycle will work, because there will be enough glyceraldehyde to have 1 go one way and 5 go the other way to finish the cycle. There'll be 6 of them. But 1 glyceraldehyde 3-phosphate is only half a glucose, which means to get a full glucose molecule, the Calvin cycle would have to run a full 6 times. Because 3 times only produces 1 glyceraldehyde 3-phosphate that's directed to making glucose. The other 5 have to go back in this cycle. So 3 times through gives you 6, but it only gives you 1 that's available for glucose. Okay? 3 times through gives you 6 G3Ps, but only 1 is available for glucose because the other 5 have to continue the cycle to go around again the next time. And so that means that the cycle would go around 3 times and make 1 G3P, then 3 more times to make another G3P, which could then be stuck together in the reverse reactions of glycolysis to give us the 6 carbon glucose. So there's a bit of an accounting there that's a little bit tricky with this cycle. Yeah. Yeah, because essentially, if you think of it this way, we're starting with CO2. So we need to effectively add 6 CO2s to get 6 carbons. Okay, I'm going to do it one more time. Yes, so we have to adjust the numbers of the NADs and ATPs to reflect this, right? So those should become 6s on this side, just very quickly. Let's just fix those numbers because we've changed the numbers to reflect the cycle going 3 times. So we're essentially multiplying everything by 3. So those would become 6s. And so would these become 6s if we run it 3 times. And then there'd be 3 ATPs on the other side, right over here. So 6, 6, 6 H pluses. And then over here, we would have 3 ADPs and 3 ATPs here. Those ATPs and NADHPHs, of course, came from the light-dependent reactions. That's where they came from. So that's essentially where this is. No, the inorganic phosphate, well, technically, yes, there's inorganic phosphate released, but it's added to this molecule to make it a bisphosphate, because before it was just 1. So you can write it in there, but then it seems to show up twice, so I just don't write it, because it's already showing up in the ribulose bisphosphate. All right.